Welcome everyone to our IGVF Scientific Seminar Series. I just want to make a quick announcement that we do have a number of open slots for presentations for this year. I'm putting the spreadsheet in the chat. We highly encourage PhD students, postdocs, staff scientists, and other early career researchers to present their work. It's a great forum to get feedback. And the talks are recorded and we do post them onto our YouTube channel. So it's a great way to get exposure for yourself and for your research. Okay, so today is a fun day because we have two PhD students who are presenting. We're going to continue with the format that the consortia voted on, which is the two 15-minute talks, followed by half an hour of discussion and questions. If you want to put a question in chat, that's okay, but we really strongly encourage you to save it until after the talks and then to speak up and ask your questions and start a discussion. So without further ado, I'm going to introduce our first speaker, which is Huijue Lyu, and she's going to discuss identification of silencers and repressive loops in AML.
Hi, let me share my screen first. Can you see my screen?
We do, thank you.
Okay, hi everyone, I'm Huijue Lyu and I'm a fourth-year PhD student from Dr. Feng Yue's lab at Northwestern. So the topic of my talk today is uncovering the oncogenic role of repressive loops and silencer hijacking events in acute myeloid leukemia. So acute myeloid leukemia, or AML, is the most common type of acute leukemia in adults with very bad prognosis. So it is characterized by differentiation arrest and uncontrolled proliferation of immature progenitor cells of the myeloid lineage, which leads to impaired hematopoiesis. So AML is highly heterogeneous with respect to the molecular characteristics. And while the genetic mutation of AML have been extensively studied, the identification of cis-regulatory elements and the 3D genome organization in primary AML patient samples are very rare. So to study the dynamic relationship between genetic and epigenetic abnormalities, chromatin 3D structure alterations, and gene expression in AML, in this work, we studied a total of 25 AML patient samples with various mutations, and 7 normal controls, including both PBMC and HSPC cells from healthy donors, and also 4 AML cell lines. So we comprehensively profiled the genomic, epigenomic, and chromatin structural changes of those samples by the assays listed here, and generated more than 200 data sets in this study. So we then extensively studied their association and the downstream functional implications. And due to the time limit, today my talk will mainly focus on the repressive loop and the enhancer silencer hijacking part of our work. So first, to understand the AML-specific 3D chromatin landscape, we performed a non-supervised clustering based on the first principle of the Hi-C. We noticed that for the samples that have similar mutations, they also had very similar patterns of chromatin compartmentalization. And next, we look at chromatin looping interactions. So on average, we identified around 200 to 300 AML-specific loops in each sample. And as shown by this example APA plot for AML-specific loops in patient 1021, those AML-specific loops are present only in the AML sample, but not in the HSPC controls. We also observed a subtype-specific pattern of looping, which involves many known AML proto-oncogenes. So one example is at the MYC-N locus. So as pointed by the black arrows, the MEK-N formed a subtype-specific interaction with a cluster of co-occurring enhancers around 650 kb downstream. So such loops were only observed in samples with TET2, CEBPA, and STAC2 mutations accompanied by active MEK-N expression. But neither the loops or MEK-N expression was observed in samples with RANKS1 or KMT2B mutations. Interestingly, we also saw that the enhancer mark H3K27 acetylation at the other anchor of the loop is only enriched in samples where the loops are present. So after we saw that some AML-specific loop have enhancers at their anchors, we then wondered whether there are AML-specific silencers involved in chromatin looping. So to answer that, we first look at the silencers in our AML samples. So we defined silencers using distal H3K27 trimethylation peaks. And across all samples, we identified more than 144,000 silencers, and on average 10,000 to 20,000 silencers in each AML primary samples. Here shows the H3K27 trimethylation tracks in 2 example regions of our identified AML-specific silencers. And as we can see, in both examples, there is an enrichment of H3K27 trimethylation in the AML patients, but not in the normal PBMCs. So interestingly, by integrating our Hi-C and cut and tag data, we found that the silencer mark H3K27 trimethylation is enriched at the anchor of a substantial fraction of AML-specific loops. So we defined those AML-specific loops with gene promoter at one anchor and H3K27 trimethylation at the other anchor as promoter silencer loops. And across all the samples, we found around 11% of the AML-specific loops were between promoters and silencers, with an average size of 169 kilobases. And we next asked whether these promoter-silencer loops have an impact on their target gene expression. So by looking at the gene expression profile, we found that for the same set of genes across different samples, they had lower expression level in patients where they were involved in promoter-silencer loops, comparing with in samples where they are not in promoter-silencer loops. So here is one example of promoter-silencer loops at the IGVF gene locus. So the IGVF gene promoter is forming 2 loops as indicated by the black arrows, with 2 silencers in the highlighted regions in several patient samples and also Kasumi1 cell line. We then performed 4C to validate these loops, and we saw high interaction frequency between the IGVF promoter with those 2 silencers. So we then further validated the loop by FISH in collaboration with Dr. Yue Yang's group at Northwestern. We observed almost spatial colocalization of the IGVF promoter and the looped silencer in the nucleus. And the distance distribution showed that the spatial distance between the promoter and the looped silencer is significantly shorter than between the promoter and the control region with similar linear genomic distance. So interestingly, samples with the silencer loops showed extremely low expression of IGVF comparing to normal controls or samples without the loops. And we also saw that the lower expression of IGVF is correlated with worse patient prognosis, and therefore we further tested the functional role of the repressive loops involving IGVF. So to validate the function of the loop, we adopted the CRISPR-I system and used dCas9-VP64 to target those 2 putative silencers indicated as S4 and S5 in this figure. One important question is that whether the looping is needed for the silencer to downregulate the target genes. So in addition to the non-specific sgRNA controls, we also targeted 3 other nearby silencers that do not loop to the IGVF2 promoter as a control. So with our system, we observed a significant reduction of the H3K27 trimethylation signal at the sgRNA targeted regions. And interestingly, we saw that targeting the looped silencers S4 and S5 reactivated the gene expression of IGVF2 by 35-folds, while targeting the 3 non-looped silencers did not change the IGVF2 gene expression. And therefore, we suspected that the gain of promoter silencer loop is a potential mechanism for silencing tumor suppressors in AML. Here's another example of AML-specific loops at the RTTN gene locus. We showed that the RTTN gene is looped to a 50 kb silencer, and after we heterozygously removed the silencer by CRISPR deletion, the RTTN gene expression was increased by 3-folds. The deletion of the silencer looped to the RTTN gene also dramatically slowed down the AML cell proliferation and also decreased the size of the colonies in colony formation assays. This further suggested that the repressive loops can play a role in AML oncogenesis. Notably, we also identified stripes that connected promoters and silencers. For example, we observed a repressive stripe for the KLF4 gene in multiple AML patient samples. We can see that the entire sliding zone for the stripe was enriched for H3K27 trimethylation signal in AML samples, but not in the HSPC or the PBMC controls. And this stripe was also associated with decreased expression of the KLF4 gene, which is an important regulator of the myeloid cell differentiation, and its downregulation also contributes to the AML leukemogenesis. So that was the repressive loop part of our work. And next, I will talk about the enhancer silencer hijacking part, and I will start with some brief background. So AML is also characterized by a high degree of heterogeneity in chromosomal abnormalities. WHO classified AML based on their structural variations and also highlighted the prognostic value of SVs in clinical settings. And here shows 8 most well-known recurrent SVs in AML. These SVs can cause formation of novel fusion genes and can also lead to abnormal gene regulation by mispositioned distal regulatory elements. So interestingly, recent works, including ours, showed that structural variations can also disrupt the 3D genome organization and cause abnormal gene regulation by enhancer hijacking events. And to further study such events, our lab previously developed computational frameworks to detect SVs from contact maps and also systematically reconstruct Hi-C maps surrounding the SV breakpoints and predict the chromatin loops induced by SVs. Here shows an example SV detected from a Hi-C of a pediatric brain tumor cell line from our previous study. We can see an abnormal interaction block between the chromosome 8 and chromosome 12, indicating the presence of a translocation event between those 2 chromosomes. And from this result, we identified a novel gene fusion event in the pediatric brain tumor. And this fusion was also validated through FISH. So here in this study, we applied the SV detection and reconstruction framework to our primary AML patient Hi-C. Here shows 2 example Hi-C contact maps from 2 different patients. And in the left contact map, we identified 2 translocation events, with one between chromosome 8 and chromosome 21, and another between chromosome 10 and chromosome 13. And in the right contact map, we identified a translocation between chromosome 9 and chromosome 22. So we then systematically identified a recurrent enhancer hijacking in our AML patient samples and cell lines. And we also asked whether these hijacked enhancers were important for the survival and proliferation of AML cells. So to answer that, we performed CRISPR-I screening using the dCas9-KRAB MECP2 system for Kasumi1 cell line. There were in total 74 new promoter enhancer loops involving 44 non-redundant hijacked enhancers identified from Hi-C. And as a result, we found that 14 sgRNAs targeting 13 enhancers were significantly depleted in the post-screening library, suggesting that they might be essential to the AML cell survival. We then performed the luciferase assays to test the activity of 5 of the candidate enhancers, and all of them increased the luciferase expression. So to further validate our CRISPR-I screening result, we next performed 6 individual CRISPR-I experiments. Here shows an example of the result for CRISPR-I validation experiment of enhancer 1, which was predicted to regulate the NSMCE3 gene. So after we disrupted the enhancer 1 with dCas9-KRAB MECP2 using 2 different sgRNAs, the NSMCE3 gene expression was reduced to 52% and 36% respectively. And by looking at the gene essentiality data from the ACLES project from the Broad Institute, we found that the disruption of NSMCE3 impaired cell growth in all the AML cell lines tested. And our CCK8 result in Kasumi1 with sgRNAs targeting the enhancer 1 confirmed that both sgRNAs caused significantly slower cell growth and proliferation. So together, this data provided further evidence that hijacked enhancers might promote AML cell expansion by increasing the expression level of their target genes. Interestingly, in addition to enhancer hijacking, we also observed that around 5.7% of the SV-induced novel chromatin loops happen between H3K27 trimethylation marked silencers and gene promoters. Such events are very similar to the enhancer hijacking events, and therefore we defined this as silencer hijacking, which was never characterized before. We also saw that the genes linked to the hijacked silencers showed decreased expression, suggesting a role of silencer hijacking in transcriptional regulation. So here is an example in the Kasumi1 cell line. We observed that an inversion of chromosome 15 brought the gene EXD1 into proximity with a distal silencer, leading to the formation of novel chromatin loops. So to functionally study these hijacked silencers, we used the CRISPR-dCas9-UTX system to de-repress the hijacked silencers by removing the repressive mark H3K27 trimethylation at the target regions. And as shown here, our system specifically removed the H3K27 trimethylation at the target regions, and the EXD1 gene expression was increased by almost 6-fold. And here's another example in the THP1 cells at the ALG10 gene locus. And the de-repression of the hijacked silencer increased the ALG10 gene expression by 2.7-fold. So together, our result confirmed that silencer hijacking is a mechanism of transcriptional regulation in AML. And in this work, we identified a total of 464 silencer hijacking events across all AML samples. And although in this study, we only look at such events in AML, we believe that this can also be an interesting research direction for other cancer types as well. So in summary, in this work, our extensive profiling of around 30 primary samples with more than 200 high-quality genomic and epigenomic datasets can serve as an invaluable resource to the future leukemia studies and also therapy development. We also uncovered genome-wide gain of repressive loops as a novel mechanism in silencing tumor suppressor genes in AML. We also systematically identified enhancer hijacking events in AML and functionally validated their essential role in AML cell survival. And this is also the first study that reported silencer hijacking events and highlighted their previously unappreciated role in oncogenesis. So in the end, I want to acknowledge Dr. Feng Yue, our lab members, and people who worked on this project, and also thanks to our collaborators who provided us the primary samples and also clinical insights, and finally to the funding sources. And thank you for your attention, and I'd be happy to take any questions.
Thank you. Thank you very much. That was a great presentation.
Thank
So we're
you.
going to have our question